welcome back to the deep dive. We are jumping straight into, well, I was going to say the deep end of the pool, but maybe it's more like the deep end of the petri dish today.
I think that's about right.
We've got this study. It's out of the University of Wisconsin-Madison, published in PLOS Biology, and honestly, it is one of the most significant pieces of astrobiology I think I've read in a long time.
It is a fascinating study.
It deals with a classic predator-prey relationship, but the setting, I mean, that's what changes absolutely everything.
Right.
We're looking at what happens to this evolutionary arms race between bacteria and viruses when you take them off Earth and stick them on the International Space Station.
Yeah, and the thing that, jumps right out at you is that this isn't just about survival. We've known for a while that things can survive in space. This is about dynamics.
Dynamics, okay.
It's about how the fundamental rules of engagement, the biological warfare, really, how that changes when you take gravity out of the equation. The lead author, Vatsan Raman, and his team, they didn't just ask, did they live? They asked, how did they fight?
Right, and we're not talking about some obscure, deep-sea vent bacteria here. The researchers chose the heavyweights of the microbiology world.
Oh, the absolute A-listers.
In one corner, you've got Escherichia coli, the K12 strain, which is, it's the lab rat of molecular biology.
It really is. And in the other corner, you've got its arch nemesis, the T7 bacteriophage.
And that's a perfect pairing for this kind of experiment, isn't it?
It's the perfect pairing. Anyone in microbiology knows T7. It's that classic lunar lander-looking virus. It's incredibly efficient, has a really fast replication cycle, and its genome is simple. It's well mapped.
So it's easy to spot changes.
Exactly. If you want to spot a mutation, T7 is your candidate because we know exactly what it's supposed to look like.
So the setup here is a classic a twin study. It kind of reminds me of the NASA experiment with the Kelly brothers, but this is all on a microbial scale.
Right.
You have one population of E. coli and T7 slugging it out in a lab in Wisconsin, and an identical population doing the same thing up on the ISS. But I want to pause here for a second because I think people might underestimate what microgravity actually does to a single-celled organism. It's not just that they're floating around, right?
No, and that's a really critical point. We tend to think of microgravity as just, weightlessness, but at the cellular scale, the physics of fluids, it all changes.
How so?
Well, on Earth, you have convection. Heat rises, cool fluid sinks, and this creates a natural mixing. In orbit, that's gone. It just stops. You also lose sedimentation, so bacteria don't settle.
So effectively, the environment itself becomes, for lack of a better word, stagnant.
Precisely. It becomes what we call diffusion limited. The bacteria are basically surrounded by a little halo of their own waste products, and nutrients don't flow to them as easily.
Wow.
So this induces a state of stress before a virus even shows up. The bacteria are already fighting the environment and the predator.
That completely sets the stage for the first big surprise in the data then. Because when the researchers let the T7 phage loose on the bacteria in space, the infection, it didn't happen on the same timeline as it did back on Earth.
Not at all. There was a significant lag, a substantial delay. On Earth, T7 is just a killing machine. It finds a host, locks on, injects its DNA almost immediately.
But not on the ISS.
In the ISS samples, that initial collision just took much, much longer. And that goes right back to the physics we were talking about.
Yeah.
If you don't have gravity mixing things up, the encounter rate just plummets.
It's like trying to find someone in a crowded room versus trying to find them floating in a pitch black void. You'll bump into them eventually, but it's going to take some time.
A long time.
But, and this is the key from the report, once they did meet, the virus was still lethal. It still did its job.
It did. The infection was successful. But something happened during that lag time. Because of the delay, because of that environmental stress, the T7 phages in space, they started to accumulate mutations.
And not just random ones.
No, in a pattern that was completely distinct from the Earth control group. They weren't just waiting around. They were optimizing.
This is the part of the study that I found a little bit unnerving. When they sequenced the genome of these space phages, they found changes in the receptor binding protein.
Yep, the molecular grappling hook.
The grappling hook the virus uses to grab the bacteria.
The tail fibers.
The tail fibers. So the virus essentially re-engineered its own landing gear. But I had to play devil's advocate for a second. Evolution is random, right? Mutations are happening all the time.
Sure.
So how do we know this wasn't just like random genetic drift? How do we know the virus was actually adapting and not just getting lucky from, say, radiation or something?
That is the million-dollar question, and it's why this paper is so significant. The researchers use a technique called deep mutational scanning.
Okay, can we break that down?
Sure.
Because deep mutational scanning sounds a little like jargon, but I know it's a really rigorous method.
It's an incredibly powerful tool. Think of it like this. Traditional evolution is like guessing a password one character at a time. It's slow, it's sequential.
Okay.
Deep mutational scanning is like a brute force attack. It's like you're simulating trying every single possible combination at the same time. They could map the entire fitness landscape of that protein.
So they could see which changes actually made a difference.
Exactly. They could see the mutations in space weren't random noise. They were all clustering in the specific areas that control binding. The virus was converging on a solution. It was finding a better key for the lock.
And the E. coli weren't just sitting there taking it.
Oh no, they were evolving too. And this is the arms race part. The bacteria in space were under double pressure, right? The weird physics and the viral attack.
So they mutated their cell surfaces.
They did. Specifically, they altered the lipopolysaccharide receptors, the LPS receptors.
And those are the handholds that the virus grabs onto.
They're the handholds. On Earth, a common defense for E. coli is to just shorten or get rid of these LPS receptors. It's like a rock climber on a wall, and the wall just retracts all the holds.
So there's nothing for the virus to grab.
Nothing to grab, no infection. This creates resistance.
The bacteria basically goes bald to stop the virus from pulling its hair.
That's a colorful way to put it, but yeah, it's accurate. Now here's the twist. The space-evolved T7 phage. It developed mutations that let it grab onto these shortened receptors.
It learned to climb the smooth wall.
It learned to climb the smooth wall.
And this is where the study goes from just cool space trivia to, oh my gosh, this matters for medicine. Because that specific defense mechanism, shortening the LPS receptors, that's exactly how some superbugs on Earth survive, right?
It is. We're talking specifically about strains of E. coli that cause urinary tract infections, UTIs.
Which are notoriously hard to treat.
Incredibly hard, because the bacteria are so good at changing those surface receptors. We've basically run out of keys that fit their locks.
But the space virus, it forged a new key.
It did. When the researchers brought the samples back to Earth, they tested the space-evolved T7 against resistant E. coli from human patients. The space virus could kill the bacteria that the Earth virus couldn't even touch.
I just want to let that sink in. We sent a virus to space, exposed it to an alien environment, and it solved a biological puzzle that we have been struggling with for years in labs down here. It sounds almost too perfect. Why didn't the Earth Control Group do the same thing? Why didn't it figure out how to grab those shorter receptors?
Because it didn't have to. The physical environment didn't force it. On Earth, with gravity and convection, encounter rates are high. The virus can afford to be, evolutionarily lazy.
There are plenty of easy targets.
Plenty. But in the diffusion-limited environment of the ISS, every single contact counts. The selection pressure was just. It was immensely higher. It's the difference between training in a comfy gym and training on the side of Everest.
That brings up a quote from Vatsan Raman, the lead author, which I thought was brilliant. He said, "We need to stop looking at space as a place to just verify what we already know."
Yes. He said, and I'm paraphrasing, that he hopes this encourages researchers to think of space not just as a place to replicate Earth experiments, but as a different physical environment that can uncover new biological insights.
That's a paradigm shift.
It really is. For decades, a lot of ISS science has been, let's grow a tomato and see if it's still a tomato. Raman is saying space is a discovery engine. It's a lab where the laws of physics push biology down paths that literally don't exist on Earth.
It's almost like we've been trying to study biology with one hand tied behind our back, and that hand is gravity.
That's a great way to put it. We never realized how much gravity was masking certain evolutionary potentials until we took it away. We are seeing biology's full potential for the first time.
So practically, what does this mean for the future of, say, pharmaceuticals? Are we going to have ISS-grown medicines?
Probably not. We might not need to grow the medicine in space, but we might need to design it there. Or at least use space to find the blueprints.
So we find the mutations up there.
Exactly. If we can identify the specific amino acid changes that let T7 kill those superbugs, we can just reverse engineer them here on Earth with CRISPR. We don't need to launch every batch. We just needed orbit to show us the design.
It turns the ISS into a high-speed evolutionary accelerator for drug discovery.
It does, and think about the range here. We're talking about E. coli, but the principle applies to Salmonella, Pseudomonas, Any pathogen where surface resistance is an issue, you could send up a whole cocktail of phages and superbugs and just see what weapons they develop.
That's such a fascinating inversion of the usual sci-fi trope. I mean, usually the fear is the Andromeda strain, right? Something comes down from space and kills us.
Right.
But here, the mutant space virus is actually the hero. It's the thing that might save us from the superbugs we created down here.
The irony is pretty thick. We're essentially outsourcing our R&D to evolution and microgravity, but we do have to be careful.
Why is that?
Well, the study also showed the bacteria got tougher. They thickened their cell membranes. They produced more biofilm precursors.
Right, let's talk about that. Because if the virus is getting stronger, but the bacteria are also bulking up, doesn't that present a risk for the astronauts themselves?
That's the other side of the coin. That's the one that keeps NASA flight surgeons awake at night.
Because the astronauts are walking ecosystems.
They are. They have E. coli in their gut, staph on their skin. If those bacteria are getting the same high-altitude training, they could be becoming more resistant, more robust, harder to kill.
And if an astronaut gets a UTI on a 6-month mission, and the standard antibiotics don't work because the bacteria have evolved space armor.
You've got a major medical emergency in a sealed can 250 miles above the Earth. And this study confirms it's not theoretical. The bacteria do change.
This seems like a massive consideration for a Mars mission. That's a 3-year round trip.
The implications are staggering. On a Mars mission, you can't just get a resupply. You're a closed loop. If your microbiome, the trillions of bacteria inside you, starts to drift evolutionarily, it could affect everything. Your digestion, your immune system, even your mental health.
And if you introduce a virus, even just a common cold, into that mix.
We just don't know what the end game is. This experiment was a simple model, T7 and E. coli. But the human body has thousands of species interacting. It's a huge unknown.
It's startling to realize how much we rely on Earth's conditions to keep us healthy without even thinking about it.
We are Earth life. Every strand of our DNA and the DNA of every microbe inside us was written by 1 g of gravity.
To wrap up, I'm just left thinking about the scale of all this. We're always focused on the rockets, the engineering, the radiation shielding. But maybe the biggest challenge is the microscopic passengers we can't leave behind.
I think you're right. We bring our biology with us, and biology is not static. It adapts. And we have to be ready for the fact that the organisms that we take to Mars might not be the same organisms that come back.
That is a really lingering thought to leave our listeners with. You know, we always wonder if we'll find life on other planets. But maybe the most alien life we'll encounter will be the life we brought with us, just transformed by the journey itself. If a few weeks in orbit can turn a standard virus into a superweapon, what kind of biology walks off that ship after the first trip to Mars? And is our immune system trained only on Earth ready for it?
That is the question we absolutely have to answer before we open that hatch.
Fatsin Rahman and his team have given us the first clue. And it is a big one.
Thanks for joining us on The Deep Dive. We'll see you next time. Stay curious.